So the first presentation will be by a student in experimental surgery named Rasai Yilmaz. So the title of Rasai's presentation will be Development and Validation of an Intelligent Surgical Technical Skills Continuous Monitoring and Assessment System. So Rasai is studying specifically technical ability and is focused on continuous real-time assessment in order to provide tailored feedback and risk detection to surgical professionals. All right, Rasai, whenever you're ready, you can share your screen.
Hello. I'm sharing my screen. Can you see my screen?
Yes.
Okay, perfect. I'm just recovering from COVID infection. Because of that, I'll be playing a video recorded previously, if that's okay.
Yeah, that's perfectly okay.
Everyone, I'll just share with the sound. Hi everyone, my name is Rasai Yilmaz. I'm a third-year PhD student in experimental surgery program at McGill University. This presentation is going to be about the Intelligent Continuous Expertise Monitoring System, the ICMS for short. Our goal at the Neurosurgical Simulation Center is to help the globalization of safe surgery through the utilization of simulation and artificial intelligence. The goal of this project was the development and predictive validation of a surgical technical skills continuous monitoring and assessment system. Simulation platforms are especially important in training those who work in high-risk situations such as military personnel and astronauts. Neurosurgery is one of the high-risk surgical specialties where roughly 1/3 of the mistakes considered technical errors. Neurosurgical simulators are developed to realistically replicate real operative procedures and have trainees practice their technical skills before ever performing on a real patient. These platforms provide vast amount of data. To give you an idea, this is 1 second of the performance recorded by the NeuroVR platform. Having such data, artificial intelligence methodologies are increasingly being used. One of the latest works from our lab provided 4 different machine learning classifiers, the best one achieving 90% accuracy in detecting surgical skill level of the performer. However, these previous applications had limitations with their post-hoc analysis. The post-hoc analysis couldn't offer real-time assessment, it couldn't track the variations happening throughout the procedure, and it had no ability to detect errors. To address these concerns, the ICMS was developed to make a real-time performance assessment, risk and error detection, and coaching to help improve performance. Data that belongs to 50 participants was used. 14 neurosurgeons and 12 medical students' data was used to train and verify the intelligence system. Once these steps are completed, data from 24 neurosurgery residents who are at different stages of training was used to assess the predictive validity of the proposed intelligence system. Each participant repeated 2 simulated tasks a total of 6 times. Therefore, a total of 300 trials were available to develop and test the intelligence system. Participants used an ultrasonic aspirator in the dominant hand to remove the tumor, and a bipolar forceps at the non-dominant hand for supportive movements and for cauterization when there is bleeding. Data from neurosurgeons and medical students were used to train the intelligence system. This system was tuned to score a neurosurgeon with a score of 1 and a medical student with a score of -1, and make this skill assessment at 0.2 second intervals. After this AI training step, the algorithms were ready to use for performance assessment. This video demonstrates the real-time assessment made by the algorithm on a trainee performance. Blue color represents more skill level, closer to neurosurgeon level, and red color represents less skill level, closer to medical student level. The colored indicator shows the real-time assessment. The decisions are recorded at graphs around in relation to different performance features. If you look at them, this person was performing at a more skilled level most of the time. However, there are time points where he was closer to less skilled level. If you look at the graph at the right bottom corner, for example, the time points where two instruments were separate were more red, indicating that this algorithm wanted both instruments used together, and whenever this person decides to use the two instruments together, he gets higher scores. The assessment is what is learned by the algorithm from the data without any human input. To make statistical comparisons, we calculated an average score for each task using the intelligence system and compared neurosurgical residents to neurosurgeons and medical students. The average score was significantly higher for neurosurgeons than senior residents, and junior residents' average score was significantly higher than medical students. Interestingly, the ICMS was also able to differentiate between senior residents and junior residents. Using neurosurgery residents' data with a linear regression analysis, average performance score correlated with the year of training in neurosurgery. The average score increased by 0.09, almost 5% per training year. In conclusion, the ICMS provided a continuous real-time assessment of surgical bimanual technical skills. The ICMS demonstrated a predictive ability on residents' technical skills acquisition throughout a neurosurgery training program. This work was accepted for publication in Nature Digital Medicine recently. For future direction to this work, we are working on the development of data recording real operative instruments. Intelligence systems can be an important part of future operating rooms to aid the surgeon in the performance of safe surgery. Thank you for listening. I'll be happy to answer if you have any questions. So if you have any questions.
Yep.
I raised my hand. I wanted to ask a question. This is amazing because you have bimanual haptic devices that work with pinching, because pinching is really difficult. And I noticed in your neurosimulation, because you have that, which is kind of fairly easy to measure the distance and the three-dimensional positioning of things. But I noticed something that it's really more advanced, which is hemostasis. How do you do that hemostasis? And my third kind of question, is this an evolution of Neurotouch? Because it looks way more advanced. Thank you very much. And it's really amazing. I'm blown away.
Yes, so yes, this simulation is this was initially named Neurotouch, and then it was updated to NeuroVR. So it's the same simulation with more advanced haptics and more advanced simulations. So in the simulated task, the bleeding was made as close as possible to real patient brain tumor. So it has bleeding effect, and in the background, it calculates all the bleeding happening from the blood vessel and from tumor and the surrounding tissue. So using that data, we were able to in another algorithm, for example, we were able to detect how much bleeding is expected so that we can give warnings beforehand bleeding can happen. So yeah, exactly, the simulation was made as realistic as possible, and it's still improving, I can say.
Yeah, thank you very much. The hemostasis part blew me away. I've never seen that before, and I've seen a lot. I wish you good luck, and I wish you well, because this is amazing. Thank you.
Thank you so much for the question.
Yeah, I agree with the video. This is really wonderful work, and congratulations. So in your title, you use the word safe. So is this safer than what are you comparing it with? Is it safer? Does it mean the other was not safe, the previous surgery?
in this analysis, so with this, the difference of this system, this AI system, is the ability to track the performance continuously. So in the previous applications, we weren't able to detect errors, for example. So we can say, we were able to say an global expertise level about the performance, but in this case, we are more specific and we can exactly tell you when you're making a mistake. Therefore, this system could be the most detailed and precise AI system to increase safety.
Okay, thanks.
All right, so are there any other questions before we move on to Dr. Chahine? Okay, it seems like that was the last question. So I will pass the torch off to Dr. Mwale, who will introduce Chahine. Thank you again, Rikai, for that really cool presentation. I think everybody really enjoyed it.
Thank you so much.
So thank you, Rikai, and thank you, Aish, for moderating. So Dr. Chahine is an associate professor in the Department of Orthopedic Surgery at Columbia University in New York, and has faculty affiliations with the Columbia's Department of Medical Engineering, Cellular and Molecular Physiology, and Biophysics program and the Columbia Stem Cell Initiative. So she directs a multidisciplinary research lab and also serves as the associate director of the Carroll labs for orthopedic surgery at Columbia University Medical Center. She also holds a bachelor's degree in biomedical engineering, which she got from Boston University. And then she completed her graduate studies at Columbia and is one of the early graduates of the biomedical engineering PhD program. Dr. Chahine completed her postdoc training as an Ernest Lawrence Fellow at Lawrence Livermore National Lab in California, where she trained in micro and nanotechnology. She started her faculty career at the Feinstein Institute for Medical Research and Hofstra Northwell School of Medicine. And she joined the faculty at Columbia University in 2017, which is her alma mater. She is past chair of the Cell and Molecular Bioengineering Council of the Biomedical Engineering Society, also past chair of the Orthopedic Research Society Spine Section, and is currently serving as a standing member on the NIH Skeletal Biology Structure and Regeneration Study Section. It is a pleasure to have you speak, and thank you very much, Dr. Chahine. Look forward to your talk. Thanks.
Thank you very much, Dr. Mwale, for the opportunity to join you all today and to share some of our ongoing research studies on disc degeneration. I'm going to share my screen. Let's go into presentation mode. Okay. Hopefully you can see this all well. So again, it's a pleasure for me to be here with all of you today, and look forward to hearing your thoughts about some of these ongoing studies. And feel free to ask questions at the end, or if you have a burning question, I'll let Dr. Mwale decide how we want to do questions. I guess I'll just say that. So as given in this warm introduction, I am a faculty in orthopedic surgery, and my background and training is in biomedical engineering. And because of that, my research is very much an encompassment of biomedical engineering type methodologies and approaches in the study of problems related to the spine, and specifically the intervertebral disc. And one of the reasons why we study this tissue and this field is because of the very high prevalence of intervertebral disc degeneration, which is a leading cause of disability worldwide. As you can see in some of this graph that I've shown you here, this is data from 2018 on the global burden of low back pain that was published in The Lancet. that showed what we had known for a long time before. So in 1990, it had been shown that low back pain was a disease that affected adults worldwide at the prime of their adulthood, unlike osteoarthritis and other degenerative diseases that come on at later ages. But what's really stunning is that in the 25 years between 1990 and 2015, there was an 54% increase in the number of disability adjusted life years that have increased because of low back pain. And so this has been a long ongoing problem, and it continues to be a long ongoing problem worldwide, and it is not getting any better, despite some efforts in both the medical and research fields. And so what I'm going to talk to you today about are some of the areas that cause low back pain, and those are related to pathological changes in the intervertebral disc. And this table I'm showing you summarizes the associations between disc-related pathologies and back pain, where there is the most evidence for changes such as disc degeneration, modic type 1 change, which is an inflammatory change in the vertebral body, the presence of annual fissures, as well as some changes related to herniation like bulging, protrusion, and extrusion. I'll show you some pictures of these kinds of conditions momentarily. So the intervertebral disc is a soft connective tissue that exists between the vertebral bones in our spinal column. It is a composite tissue with a gelatinous nucleus pulposus in the center and a fibrotic annulus fibrosus in the periphery, shown here in a cross section. And there is a thin cartilaginous end plate layer that plays an important role in terms of diffusion and nutrient transport, because the intervertebral disc is in fact the largest avascular organ in the human body. And so with disease and aging, there are changes such as thinning of the height of the intervertebral disc. There could be some mild bulging. There could be total extrusion or herniation of the nuclear material, this gelatinous material. There could be fragmentation and degeneration of the tissue, and there could potentially be the formation of osteophytes, which are pathological bony formations in soft tissues that formerly did not have bone formation. And that could also occur in facet joints in the spine. And so when we look at what happens with the intervertebral disc, what you see is that there is loss of compartmentalization of these two regions that have very different functions. And therefore, it sort of becomes a more of a fibrotic tissue that has very different matrix as well as mechanical function. We also see this formation of these bridging osteophytes as well as change in disc height. Now, one of the reasons why this can be a painful condition is because of the anatomical location of the spinal canal and the spinal nerve roots that pass posterior to the intervertebral disc. And so pain can be generated potentially by that causing stenosis of the spinal canal, or by actual impingement of a spinal nerve root by a herniated disc, as well as some other mechanisms that are still under investigation in the field about why a disc that does not cause stenosis or herniation can also be painful. There have been many studies in the field on changes of the intervertebral disc and the extracellular matrix. I would say the extracellular matrix studies are perhaps the most long-standing findings in the field. And it is well accepted that there are changes in hydration. There's a loss of hydration and loss in proteoglycan content, matrix fibrillation. And this is thought to occur through activation of catabolic enzymes like ADAMTs and ADAMTSs. And these biochemical changes often are associated with physical changes like the loss of disc height, this convergence of the inner and outer regions, and altered matrix stiffness, and therefore load distribution. I want to bring to your attention what I found to be a very nice, elegant new publication in the literature, this proteomic study on called DIPPER, a spatiotemporal proteomics atlas of human intervertebral discs for exploring aging and degeneration dynamics from 2020. And I think one of my favorite sentences in this paper sort of summarizes in a very elegant and comprehensive data analytics way what has been sort of understood for quite some time, that the most important biological property for a young disc is structural integrity. And this is shown here with this elegant proteomics data where you see that the categories that are most expressed in these gene ontology or protein ontology, because these are proteomics data, are matrix components that are important for the integrity of the extracellular matrix. And then with aging, and in this case, these are just age tissues that are not necessarily disease tissues, what appears to be happening is that there is a loss of the matrix integrity, as evidenced here in these photos, but also as evidenced by the data where you see increases in cellular stress responses, an ever continuing matrix remodeling, as well as some inflammatory changes that are taking place in an aged but not necessarily diseased tissue. Taking this a step further, other proteomic studies in the literature, now show that matrix changes not only happen with aging, but in fact, there are other categories of changes in the proteome that are related to degeneration in of itself, that are independent of the aging phenotype. And those appear to be related to inflammation and what is termed as inflamma-aging in this study, where specifically degraded intervertebral discs, disease discs, show a profile rich in oxidative stress, apoptosis, innate immune signaling, as well as inflammatory complement activation and defense responses, suggesting that one of the main differences between healthy aging and disease are the role of inflammation and immune system processes within this avascular tissue in of itself. And in fact, there's long been bodies of work summarized in what I point to you here are 3 well-written review articles on inflammation and intervertebral disc degeneration. And broadly, what the field finds is that there are increased levels of numerous pro-inflammatory cytokines like IL-1 beta and TNF-alpha and others that promote matrix degradation. My lab has studied systemic inflammation and found that in fact there are elevated levels of circulating cytokines in patients who have painful disc disease. And these cytokines are important for a number of reasons. Among them are, as I mentioned, the matrix degradation, but also because they act as chemotactic factors to promote recruitment of immune cells such as macrophages or T cells, which then further advance the degradative profile. And it is also thought that these cytokines are important because they play a role in promoting nerve ingrowth that is pathological, which may be one of those mechanisms that I alluded to earlier for the causes of discogenic pain that does not necessarily have a specific change in stenosis or nerve compression. And so if we think about it like this, if the inflammation is a key root to many disease mechanisms like immune cell recruitment, nerve ingrowth, and matrix catabolic changes, which alter the loading on the spine, and all of these can individually or in combination promote pain. Can we use anti-inflammatory drugs to control inflammation and therefore have a way forward to dealing with disc disease and pain? And I would say that the answer to this question is mixed. And I would, as of right now, I would say probably not. We are not at a point where the field has classic, well-accepted approaches for using anti-inflammatory drugs for the treatment of disc pain and disc degeneration. However, the treatments that are out there do allude to us some potential possibilities to move forward in. Currently, clinical treatments for these kinds of conditions include bed rest, oral NSAIDs, anti-inflammatory medications, painkillers. However, these have a high risk of opioid addiction, as well as overdosing and potential death, physical therapy and conditioning, epidural steroid injections, or nerve root blocks. However, I would argue, and the literature supported, that many of these non-operative and these operative treatments are somewhat varied in outcomes and unpredictable. For example, some of these operative treatments are well indicated, such as discectomy or decompression for a herniated disc. And this can be done with or without spinal fusion, depending on the need for integrity of the tissue. However, there are some cases where spinal fusion is done without a discectomy, and those for what is called as a black disc. And the findings on those studies are very are highly varied, and the efficacy is unclear. Moreover, there are other side effects, such as adjacent segments in the spine, that then are affected with these kinds of treatments, which affect the disease progression. So given the treatment variation and unpredictability of existing therapies, it is somewhat not surprising that the onset and the preponderance of disease and its impact has not been under control or has been diminished in any meaningful way in all these years. There are recent and ongoing clinical trials looking at anti-inflammatory therapies, such as the ones I listed here. However, again, the feeling is that these results are mixed, with some studies showing short-term benefits of pain relief or return to work, while other studies don't show these benefits. And so there are inherent limitations related to many factors, among them the drug delivery approaches, the choice of therapies that are used, the patient selection, as well as study design. And so we still remain at a point in the understanding of the degenerative cascade where new insights into disease mechanisms as well as therapeutic approaches are still highly needed. And so to this end, my lab studies multi-scale mechanobiology and uses the concept of mechanobiology as it applies to cells and tissues to try to come up with new concepts and new possibilities of understanding the disease and hoping to offer some way forward in regenerative therapies or in therapies to try to reverse the onset of disease. And so in some of our more cellular work, we study cell biomechanics and the role of cell cytoskeleton and how that affects the response of the cells in the intervertebral disc. We translate some of our findings and evaluate it in organ culture models, where we study the response of rodent disc to various therapeutics. We also have some studies on in vivo therapeutic studies, again in the rodent model, looking at stem cells or other cellular biological therapies. And then as a person who's led my research career entirely in clinical departments, I've been fortunate to work alongside clinicians to study disease profiles in patients and trying to come up with potential biomarkers to help inform who's a good patient for various treatments. So what I'm going to talk to you a little bit about today is some of our work on innate immunity, and specifically our focus on toll-like receptor 4. Toll-like receptors are well studied in the context of inflammation and infection in the immune system. They are a receptor that responds to both exogenous and endogenous ligands in the immune system, and plays a role in important conditions such as sepsis and other infectious conditions. Endogenously, what it responds to are ligands related to matrix fragments like hyaluronic acid fragments, fibronectin fragments, heat shock proteins, etc. And one of the best exogenous ligands that is well studied to simulate activation of TLR4 is lipopolysaccharide, or LPS. And in most cells, either ligand can activate this a pro-inflammatory cascade of TLR4, primarily mediated through the NF-kappa B pathway, which then turns on either pro-inflammatory cytokine signaling or antimicrobial peptides, depending on the type of ligand that it's activated. When my lab started doing this work, there wasn't that much known about toll-like receptor 4 in the intervertebral disc. And so what we started out to do was to first evaluate the presence of this receptor in human IVD cells from the nucleus pulposus, which is this gelatinous central tissue here. And so you can see that NP cells, in fact, express toll-like receptors readily. And when you take these cells and you stimulate them with lipopolysaccharide, or LPS, you see robust increases in numerous pro-inflammatory cytokines like IL-1 and IL-6, but also matrix degrading enzymes like MMP1, suggesting that these kind of stimulant can mimic what is a simulated catabolic and inflammatory cascade. When we compare the response of human NP cells stimulated with LPS to a more a natural or endogenous ligand like low molecular weight HA, we indeed find that there is activation of catabolic pathways like MMP1. However, clearly the magnitudes are lower with low molecular weight HA compared to LPS, which is very potent. To confirm that these signaling pathways are specific to toll-like for TLR4, we've done some studies where we've blocked TLR4 signaling, either using a neutralizing antibody that decreases nitrite release in response to LPS, or with small molecule inhibitors like TAC242, where we see a decrease in nitrite release due to low molecular weight HA stimulation. Since we've started this work, there had been some studies by other groups that have, in fact, shown that TLR4 expression increases with disease severity. So this is data from the literature that shows that the levels of TLR4 increase with increasing degree of degeneration. And in fact, we became interested in studying a particular endogenous ligand of TLR4, which is this damage-associated molecular pattern or cytokine called HMGB1, high mobility group box 1, which we also found increases with disease severity in patient samples, just as other pro-inflammatory cytokines like IL-6 similarly share that pattern. Now, HMGB1 is a non-histone nuclear protein. It is typically present in the nucleus of the cell, and it serves a function of stabilizing DNA. However, it can be released from the cells, and it has a very different function, where then it acts like a cytokine or chemokine in stressed or necrotic cells. And so therefore, it's considered a damage-associated molecular pattern that when it's released from the cell, it acts in this pro-inflammatory cascade. And it can activate NF-kappa B pathway to promote pro-inflammatory signaling, cytokines, and MMPs, as well as other factors. So indeed, to study this in nucleus pulposus cells, what we showed here in this paper from 2019 is that we took human NP cells from healthy sources and found that HMGB1 increases both IL-6 and MMP-1 secretion in a dose-dependent manner. And when we looked at which receptors HMGB1 is signaling through in response to this stimulation in human cells, we took these experiments and blocked it with either a RAGE inhibitor, a TLR2 inhibitor, or a TLR4 inhibitor, and found that in fact the TLR4 inhibitor was the one that is most potent at blocking effects of HMGB1. We did further studies, in fact, and that's not shown here, but it's presented in the paper, where we co-stimulated with multiple receptor inhibitors and found that in fact that only when you have TLR4 inhibition in the experiment do you then see a complete inhibition of these pro-inflammatory and catabolic pathways of MMP1 and IL-6, or levels, I should say. Moreover, excited about these findings in an in vitro setting, we then went on to look at what happens when you deliver LPS into an intervertebral disc. And so to do this, we have gone to the rodent model, where we do an injection of a small volume of LPS into the intervertebral disc using a small 33 gauge needle. And the reason I mention that is because the size of the needle, in fact, impacts whether you cause disruption of the matrix independent of what you deliver to it. So you can potentially cause damage just by injecting saline. And so we've minimized this by using these small needles in the rat tail disc, and looked at outcomes up to 28 days after injection. And what we've seen is that over time, in the first 2 weeks, looking here at histological changes, we start to see fragmentation of the nucleus pulposus and compaction of the NP as a sort of surrogate for some of the early disruptions in extracellular matrix changes of the intervertebral disc. We've also done some biochemical assays that have shown for there to be pathological fibrotic matrix deposition in the NP, which I don't have here in the interest of time. However, we've looked at also biomechanical properties of these discs and found that in fact with the dose response, we see significant decreases in the compressive properties of the intervertebral disc. Excuse me.
Mommy, we got something for you.
Thank you. Can you go?
Wait, nah. No, I picked it up.
I'm so sorry about that. That's my little one coming back home. Let me reshare my screen. Intrigued by these changes in the biomechanics, where we see at the highest dose a decrease in the compressive creep modulus of the discs that receive the highest dose of LPS, which is 100 micrograms per mil, compared to sham or controlled discs that receive no injection. Moving forward from there, we began assessing whether there are behavioral changes in this model indicative of a potential pain behavior. And because we are working in the rat tail, which is not an area that is well studied from a pain behavior perspective, we evaluated a number of different testing modalities, if you will, to evaluate responses of the rodents. And one of the ones that is most promising so far is this test called pressure algometry, where we use a calibrated force transducer applied by a trained examiner onto the region of the injury to determine the sensitivity to the application of force. And so what we find here is that At baseline, our animals are very similar in their, our two groups in their ability to respond to a force. And when surgery is performed between baseline and day 1, you see that there is this decrease in sensitivity in both groups. However, the sham group recovers its pain behavior back to baseline, whereas the LPS group, at least out to 28 days, does not recover, and in fact continues to have deficiency and therefore additional sensitivity to applied mechanical stimuli up to 28 days. We examined other tests that are more commonly used in lumbar studies or other animal models, such as in the arthritis community. And we found that there are a few other findings, such as the tail flick, which is a thermal stimulus that is performed distal to the injury site at the tip of the tail. And we found some short-term changes in sensitivity. However, these are not long-lived findings. Similarly, looking at von Frey filament, which is responses in the paw indicative of a radiating response, we see some early changes at 3 days. However, these changes are not long-lived. So in fact, we're really excited about these findings with the pressure algometry that might suggest to us that we can potentially track over time axial changes in mechanical sensitivity at the site of injury in this animal model. So to summarize this part of the work, what we've seen is that TLR4 activation by LPS, low molecular weight HA, or HMGB1 triggers expression and secretion of multiple pro-inflammatory cytokines consistent with catabolic pathways of disc degeneration, and that in vivo intradiscal activation of TLR4 can initiate and potentiate the loss of matrix integrity, biomechanical disruption, and induce mechanical sensitivity consistent with localized discogenic pain. Interestingly, we find that disulfide HMGB1, which is the isoform that we've been studying, is mediated primarily by TLR4 signaling in NP cells. And in fact, that TAC242 is an effective small molecule looking at blocking TLR4 signaling in IVD cells. Moving forward, we're interested in looking at specifically the contributions of HMGB1 in the disease mechanisms of degeneration. And so fortunately, we've been able to acquire a mouse model that knocks out HMGB1, which we are doing specifically in the intervertebral disc. And we have some ongoing studies that hopefully in the next couple of years, we'll have more data on to tell you about how removing HMGB1 may be protective of inflammatory changes in response to injury, and that we hypothesize that this controlled inhibition can protect disc integrity to injury. Another part of my lab, as I mentioned, studies cellular changes in response to loading. And just to kind of put us all on the same page, I included this schematic, which is the mechanosensitivity or the mechanical environment of the intervertebral disc, where you see here that the intervertebral disc undergoes numerous types of mechanical loading, from axial compression, which is the most common or the greatest magnitude because of the loading of the spine in when we're standing up. But also this axial loading due to body weight and motion puts other kinds of forces on the disc, including biaxial compression in the annulus fibrosus, tension on the annulus fibrosus, osmotic pressure and hydrostatic pressurization due to the fluidity of the nucleus pulposus, as well as shear loading on the annulus fibrosus region. And in fact, it has long been known and well accepted that the response of cells to stimuli is in fact really critical for biosynthesis of the cells, metabolic activity, as well as maintaining the cell phenotype. And that there are numerous cell mechanotransduction modalities that the cells sense these different kinds of loading, from tension to compression to osmotic pressure. Osmotic pressure is a really interesting modality of loading, and perhaps a little less studied than axial compression or tension on the annulus fibrosus. It changes significantly with different activities throughout the day. So there has been long-standing studies that had shown that your posture affects the magnitude of hydrostatic pressure in the disc, and that in fact, hydrostatic pressure magnitude and frequency affect cell metabolism and matrix deposition. And with disease and degeneration, we know that the loss of proteoglycans in the tissue causes a loss of pressurization in the intervertebral disc. Therefore, we don't know if, for example, the cells in the intervertebral disc are responding differently because of this changes in the loading magnitude, or whether there are changes in the mechanotransduction pathways that are mediating this response. And so when we wanted to start to look at these kinds of questions, we turned our attention to this bioreactor system, which allows us to do a more high throughput studies using multi-well system and this bioreactor chamber. And so what we do here, this is a system that we acquired from the company Instron, where where samples, they could be cells or they could be tissue explants in culture, are plated in a multi-well plate and encapsulated in this chamber, which then is controlled. The pressurization of the fluid and the air is controlled by a servo stimulator, which is connected to an external air compressor, which has an input of air from the incubator. So it's taking incubator air, putting it through a compressor, pressurizing it, forcing it through the stimulator, and having a controlled compression applied to the fluid that is inside the chamber. And using this approach, we studied changes in some gene expression of NP cells and found that in fact, aggrecan gene expression increases with increasing magnitude of applied hydrostatic pressure. This is applied cyclically at 0.1 hertz for 1 week. However, things like cell phenotype markers like keratin 19 and NCadherin, in fact, are not optimized at these highest magnitudes. And in fact, they have elevated levels compared to unloaded cells at these more intermediate and yet physiologically relevant magnitudes of 0.1 megapascal. Interestingly, when looking at catabolic results or genes, we saw that there are decreases in MMPs. However, these were not statistically significant with increasing magnitude. And we saw dose-dependent increases in other mediators of inflammation like nitric oxide release. So those kinds of findings really set us up well to begin looking at this new concept that we're really focused on currently, which is evaluating the response of other cells that are part of the disease mechanism. And our attention lately has turned to immune cells, specifically macrophages. So we know from the literature that macrophages are present in degenerated and herniated intervertebral discs, suggesting that these phagocytic cells may play a critical role in pathophysiology of the intervertebral disc degeneration. And in fact, in herniated disc, there is evidence of these exogenous macrophages infiltrating the disc, and that's provided through tracing studies in mouse models. And so what was known about macrophages is that they can take on a significant different cellular phenotype, ranging from a pro-inflammatory phenotype that results in tissue breakdown to a more anti-inflammatory phenotype which can result in or support tissue remodeling. And there are different kinds of modalities for pushing macrophages towards an M1 or an M2 phenotype. And the best studied version of these modalities are the chemical stimulation using chemokines and cytokines like LPS and interferon 1 gamma, primes unstimulated macrophages to take on an M1 phenotype, whereas factors like anti-inflammatory IL-4 and IL-10 or IL-13 promote this M2 phenotype. There have been studies in the literature that also show that mechanical factors like substrate stiffness, cell shape, and mechanical strain also prime macrophages to take on a different phenotype. And so what we really know is that macrophages are known to be mechanoresponsive, and their polarization or phenotype changes towards a spectrum that is dictated by the microenvironment and chemical stimuli. And so with that in mind, and taking on this bioreactor system that we had in our hands, what we decided to do is begin to ask the question of what happens to macrophages when they are enter this mechanically loaded microenvironment of the intervertebral disc, and specifically asking about the contributions of a disc biomimetic hydrostatic pressurization. And so to do this, a graduate student in my lab, Kevin Burt, isolated bone marrow from mice and generated either undifferentiated macrophages or pro-M1, pro-inflammatory or anti-inflammatory macrophages. He took these subsets of cells and put them back in the bioreactor that I described earlier, and applied different magnitudes of loading from a healthy magnitude, which is the higher magnitude, to a lower magnitude, which is the intermediate to severe degeneration, because as I described, we see a loss of pressurization with degeneration. And then he also looked at various timing, looking at 1 versus 3 hours or multi-day loading to optimize the loading configurations. And there are a number of outcomes that he's looked at, from flow cytometry and gene expression to determine cell state or cell phenotype, transcriptomic analysis with RNA sequencing, as well as some functional assays, specifically looking at phagocytosis, and most recently looking at potential mechanistic studies to try to help us understand how these macrophages are responding to hydrostatic pressure. And so what he showed initially is that hydrostatic pressure upregulated M2 markers at higher loading magnitudes compared to M1 markers. And so you see here, with increasing magnitude of pressurization, you have arginase 1 increases, YM1 at the highest loads, and EGR2. These were M0s, undifferentiated cells that have taken on increased anti-inflammatory expression just due to the application of hydrostatic pressurization. When he took these cells and applied 3 1-hour loading cycles compared to a single 1-hour loading cycle, he saw even further increases in the anti-inflammatory cytokine IL-10. And when he compared the response of M0, M1, and M2 cells, he found that M2 cells can be further stimulated from having anti-inflammatory function by pushing arginase 1 with applied physiological hydrostatic pressurization. Moving from there, he performed some RNA sequencing to understand what are the pathways and mechanisms that these cells might be responding with. And you can see here, have an appreciation that there is an increasing number of differentially expressed genes with increasing magnitude of pressurization. And in fact, that this, the 0 and the 0.6 megapascal groups have very different profiles, biological profiles that differentiate the two groups. Looking more deeply at the specific pathways that we found to be differentiated in with hydrostatic pressurization, Kevin found that immune function regulating pathways are overrepresented at the higher hydrostatic pressurization. So in 0.6 megapascal, we see increases in pathways related to cytokine signaling, interleukin-10 signaling, interferon gamma signaling, as well as interleukin-4 and 13. So suggesting that there are some anti-inflammatory mechanisms that are at play, as well as some other pathways that are certainly novel to macrophages signaling. And interestingly, what's downregulated equally are some other interferon signaling mechanisms, as well as NTRK mechanisms. Breaking this down in this heat map, specifically, he saw, as I implied, changes in IL-10, 4, and 13 with increasing magnitude of pressurization, as well as some non-canonical TGF-beta signaling, which we believe to be an anti-inflammatory mechanism as well. Taking this a step further, he looked at IL-10 in these different profiles and found that not only does the gene expression increase, but in fact, IL-10 release into the media can be promoted and enhanced with the application of hydrostatic pressurization of M0s. So now these are cells that are not chemically primed, but rather having an active release of IL-10 into the media, acting in an anti-inflammatory mechanism through the application of a physiological magnitude of hydrostatic pressurization. However, this does not happen when you have decreased pressurization equivalent of a pathological magnitude. To see if this has any impact on the function of these cells, he evaluated the function of the cells to phagocytose apoptotic cells, and this is a condition called ephrocytosis, where he took fluorescently labeled Jurkat cells and co-cultured them with his macrophages that were treated in unloaded conditions or hydrostatic pressure conditions to determine if the cell's ability to phagocytose is different due to hydrostatic pressurization. And in fact, you could see here that hydrostatic pressurization increased the uptake of apoptotic cells early on, within 1 hour, beginning to show an effect, and a more significant effect at 3 hours post-loading. However, it appears to be somewhat short-lived. So by 24 hours, this effect is not present. And to try to understand what are the mechanisms that are mediating this mechano response of the macrophages, he began looking at some mechanosensitive pathways related to the cytoskeleton. And so here is some staining that he did on ICAM1 and found that in fact, you see significant increases in ICAM1 protein expression at 3 hours and 24 hours post-loading. And similarly, the mechanosensor YAP, which is part of the YAP-TAZ mechanosensation mechanism, also is elevated in these cells that are loaded at 0.6 megapascal at 3 hours, and suggesting that the nuclear staining is elevated, which implies that there is activation of transcriptomic pathways related to YAP signaling. So to summarize this part of the presentation, what our data shows is that macrophages are responsive to hydrostatic pressure magnitude, and that increasing hydrostatic pressurization promotes expression of anti-inflammatory phenotypes such as M2 markers. Loading also promotes well-established and novel anti-inflammatory pathways such as IL-10 secretion, and that there's evidence now that these findings sort of point to the fact that decreasing hydrostatic pressure magnitude with this degeneration, which may be due to the loss of tissue hydration, may potentially promote a loss of an anti-inflammatory phenotype of resident or recruited macrophages And we're, thinking about ways that we can try to test that as a possible disease mechanism, that the change in pressurization may in of itself push resident or recruited macrophages into a more inflammatory and less anti-inflammatory or less resolving phenotype, which then may propagate or promote the disease outcomes. And so we have ongoing future studies looking at the mechanistic roles of ICAM1 and YAP to determine if they are directly mediating these anti-inflammatory responses that we are seeing in the loaded macrophages. And the last part of my presentation today is going to talk a little bit more about some of our work on the cytoskeleton, so sort of going down in scale to the cellular responses. And I wanted to sort of reflect with you that it is well known and well appreciated that cell shape is an important aspect of cell phenotype. And so in the intervertebral disc, the nucleus pulposus cells in many different tissue species are rounded. And so these are porcine and P cells that you see here. These are images of bovine NP cells. Human NP cells have some roundedness as well, as you could see here when they are in the normal histological phenotype. However, classic studies in the literature have pointed to the fact that when you have degeneration, there are these cytoplasmic extensions that appear more prominently with degenerate tissue, as well as these perhaps more pathological morphologies termed here as a stellate morphology, which are not indicative of the normal physiological phenotype of the cell. We were intrigued by these findings and wanted to ask whether these onset of this change in cell morphology is in some way related to the inflammatory milieu that these cells find themselves in with degeneration. And so what we did at the beginning of this work is we began working with the bovine model, which we feel is a really great model to recapitulate healthy human-like cells that have a similar phenotype and similar extracellular matrix to human compared to, say, rodent or porcine. And so what we did here is we isolated NP tissue from bovine lumbar spines, isolated the cells, and suspended them in alginate beads like the one you see here, so that the cells can maintain their rounded morphology, but that we can have control over them in an in vitro setting so that they don't spread and attach to tissue culture plastic. And then a parallel to this, we began studying nucleus pulposus explants and their response to inflammation and cellular response to inflammation as well. However, one of the issues in the field related to studying explants is that the presence of proteoglycans in the tissue attract tremendous amount of water, and therefore you have huge magnitudes of swelling, which cause cell death and other complications. And so in order to try to overcome those potential limitations of studying explants, what we did here is we cultured our explants in what we call the swelling restricted environment, where we took punches of NP tissue and put it in an agarose around it, cast it in the tissue culture well, and compared its response to, say, a free swelling where you only put media on it. And in fact, we showed that when you restrict the swelling using agarose, you nearly obliterate the release of nitric oxide into the media compared to free swelling, which shoots up immediately within 1 day of culture. And you significantly decrease the potential of GAG loss into the media, which is a degenerative response. We've also looked at cell viability and find that the cells are viable in this culture model up to 14 days. Using these kinds of systems, what we've done since is then taken these cells and exposed them to different inflammatory stimuli and looked at the response using atomic force microscopy, where we go down and indent a single cell, and using laser measurement techniques can backtrack the stiffness of the individual cell. We looked at hydraulic permeability using microfluidic systems, and we also looked at changes in cell morphology and cell behavior of different proteins using imaging flow cytometry. And so what we found is that inflammation here modeled with stimulation with TNF alpha, although we've since studied responses to LPS and TLR4 as well, and published that last year in the ECM journal, and where we see similar responses. So here I'm just focusing on the responses to TNF alpha. And what we find is that actin cytoskeleton is the most disrupted in response to a short TNF alpha stimulation, as evidenced here by this formation of these punctate structures of cells cultured in the alginate system. When we looked at other changes like tubulin or vimentin, we see very little changes that happen compared to the changes in the actin cytoskeleton. When we quantified the change in cell morphology using the imaging flow cytometry, we see that the cell circularity decreases. And to confirm that these are changes specific to the actomyosin contractility, we measured the expression of phosphomyosin light chain and found that to decrease significantly. And extending these kinds of findings into further transcriptomic analysis, we identified numerous genes related to myosin, Rho family, kinases, cytoskeleton, and actin-related proteins whose levels are either significantly decreased or increased in response to TNF alpha stimulation. Taking a step further to look at the biomechanical properties, we find that the cell radius significantly increases in TNF alpha. The hydraulic permeability, which is the ability of the cell to allow water in or out of the cell under swelling or shrinking conditions, that permeability to water also increases. And the modulus, the stiffness of the individual cells, decreases. And so this tells us that there are significant changes at the single cell level, which has, in my mind, significant consequence on the ability of the cell to transduce mechanical loading and mechanical forces. So to try to understand the consequences of these changes in the myosin light chain and actomyosin contractility, we asked whether using a drug like CNO3, which activates or turns on constitutively activates Rho A, whether that can potentially overcome the decreases we are seeing in PMLC, and is that mediating the biomechanical changes we are seeing in the cells. And so what we did is we co-treated the cells with TNF alpha and CNO3. And what is remarkable is that the cell shape is significantly protected from the effects of TNF alpha when you put CNO3, or meaning you increase the actomyosin contractility of the cell. Even in CNO3 alone, in the absence of TNF alpha, we see increases in cell circularity. Moreover, when we did this in our organ culture model in the swelling restricted system, TNF alpha, shown here at the top row, causes major polarization and formation of some extensions of the cell. However, when we co-treat the cells with CNO3, we can completely preserve the maintenance of this rounded phenotype that is altered in response to TNF alpha. Looking further at the cytoskeletal and Rho pathway and myosin pathways, we find that numerous targets that were upregulated in response to TNF alpha are now downregulated, and vice versa, so that the co-treatment with CNO3 is protective of changes in the cytoskeletal transcriptome, as well as changes in the single cell biomechanical properties. So here we see the hydraulic permeability decreases in response to CNO3 co-treatment, as well as the cell elastic modulus is recovered in response to co-treatment with CNO3. Excited by these findings, we wanted to take this a step further and ask if increasing actomyosin contractility is protective of cell mechanobiology and cytoskeleton. Does this have an effect on the surrounding extracellular matrix? And so again, using the organ culture model that I described to you, what we found here is that in fact, co-treatment or treatment of explants with TNF decreases the GAG content and Alshin blue staining indicative of proteoglycans in the tissue, and that co-treatment with CNO3 increases or protects against the loss of proteoglycans due to TNF alpha stimulation. And consistently, we see decreases in numerous MMPs, which are activated by TNF alpha as in response to the co-treatment of CNO3, as well as ECM genes that are affected by TNF alpha. Their levels are responsive to the co-treatment of CNO3. And to confirm whether or not these factors are happening or these protections are happening through NF-kappa B signaling, which is the presumed master regulator of degradation in response to TNF alpha, we looked at NF-kappa B nuclear localization in these cells with and without CNO3, and shown here with the imaging flow cytometry and quantified here. What you see is that in fact, co-treatment with TNF CNO3 causes nuclear translocation of NF-kappa B to be blocked back down to basal levels. And again, shown here in a more classic way using Western blot analysis where P65 levels are significantly increased in the nucleus with TNF alpha, and that co-treatment with CNO3 prevents the nuclear localization of P65 in NP cells. And similarly, numerous cytokines and other NF-kappa B responding genes are then affected both with the treatment of TNF alpha and whose levels are significantly reversed or downregulated in response to CNO3 treatment. So we are very excited about the possibility here of using what we're considering to be a more mechanobiological mechanisms for protecting integrity of cells and tissues in the intervertebral disc. And moving forward, we're thinking about ways that we can potentially deliver these kind of therapeutics into the disc. And given the avascular nature of the disc, as well as the low ability of therapeutics delivered into the disc to be retained, we're now working with a collaborator and colleague from Northeastern University on developing a drug delivery system for the intervertebral disc using a protein called avidin. And avidin is really interesting because it is a positively charged nanostructure, which we believe can be used to promote retention in the intervertebral disc next to a negatively charged matrix, which is the native matrix of the disc. So using electroneutrality, we're hoping that the negative matrix and the positive avidin molecules will attract each other and therefore cause there to be long-term retention of the drug delivery system, and therefore a drug conjugated to it for an extended duration of time. And in fact, using some ex vivo studies, we've shown in a proof of concept, in fact, that avidin can be retained into the disc with a one-time injection up to 14 days in comparison to say a small FITC molecule, which can only last up to 4 days in the disc, or a neutral but large molecule like Neutravidin, which has a shorter retention compared to Avidin, which is using its charge effects to promote retention. We've also compared the responses of NP explants that have been treated with enzymes to degrade them, to make sure that if loss of proteoglycans due to degradation inhibits or interferes significantly with the function. And in fact, we find that we can extend this retention out to 28 days, even in a degraded extracellular matrix. One last thought to leave you with. We're interested in understanding if these cell mechanobiology changes are also present in human samples. And so we are working with our surgeon colleagues here at Columbia to collect patient samples and do this detailed mechanobiological analyses on patient cells and comparing them to healthy cells or responsive healthy cells to inflammatory cytokines. And the data is promising so far, though these are early days, where we are seeing some changes in the permeability and in the modulus of the cells on the few samples that we have looked at that are consistent with our findings on the bovine model. So to summarize my talk, what I hope I've convinced you of today is that there are important crosstalk between the cell mechanobiology and the role of inflammation in disc degeneration, from innate immune signaling to immune cell phenotype, which is affected by mechanical loading, to the response of the cell cytoskeleton and contractility in response to inflammation, and how that offers a potential pathway for blocking degeneration. And so when I started my talk, I gave the premise here that, are we able to use anti-inflammatory drugs to block inflammatory cascades and the downstream effects of the disease? And while the answers to these questions are still unknown, what we believe we have uncovered in the meantime is that there are additional controls to this problem that were previously unknown, namely that immune cell mechanobiology is in fact an important aspect of response to inflammation, and perhaps we can think about this as a microcontroller of inflammatory response, that actomycin contractility can be a controller, and that in fact applied loading can have responses and effects on the matrix itself and the mechanical properties, and that perhaps we should think about how we control the mechanical environment of a degenerated disc. So with that, I want to thank the many trainees present and past who have been instrumental to the work. Timothy Jacobson and Paula Hernandez did the studies on cell mechanobiology and actomycin contractility. Kevin Bird is a graduate student who's working on the macrophage responses to loading and some of the mouse models, which I didn't get a chance to speak about today, related to HMGB1, and as well as other students who have contributed to the earlier studies on toll-like 4 receptors, like Bronte Shaw, and a current student, Lauren Leszewski, who's working more on the nociceptive question of the model, as well as our wonderful collaborators and colleagues, Clark Hung and Ron Lehman, who are both in the orthopedic space, and Han-Rui Zhang, who's a new colleague on macrophage biology, and my colleague Ambika Bajpai, who is our collaborator on the Avidin studies. Of course, I want to acknowledge our funding sources and thank you for your attention, and I'm happy to take questions.
Thank you very much, Dr. Chahine. This is a brilliant work. Loved it, loved it. Really learned a lot. Thank you very much.
Amali.
Yes, really wonderful. Questions?
Yeah.
Yes.
Dr. Chahine, I just realized how close I am to dementia after I saw your presentation, but how much I don't understand. what's happening in there. So that's that. But there are some things that are really interesting, and I really love them. For example, one of the things that I think it's interesting is that we're using finite element bio, that it's developed actually at Cornell by Dr. Atinician, I think. When you're using those models, and we have a model on that, and maybe we can collaborate on that thing. But I want to ask you, when you are using the AFM, the atomic force microscopy, which basically is like I'm using a pickup piezo element, and I'm just going over a cytoskeleton. If I noticed that you have a lot of granularity and sensitivity when you do that on applying the pressure, because I did that, and it was I was very rough on that, and I needed kind of like really hard stuff. But you are kind of like very gentle doing it, and you're able to kind of pick up the gentleness, and that will blow my mind away that you can do that at those dimensions, because we're talking about 1 micrometer. How do you do that?
Thank you very much for your question. So we are using a biological AFM system that is designed to, or I say optimized to work well with with biological materials. Although AFM as a technique certainly has the capabilities to do, single nanometer perturbations on surfaces, those kinds of level of control does not work well on biological materials. So for example, if you try to do an AFM on, glass or ceramic or some, metal, whatever, other kinds of materials, you can get very exquisite sensitivity that doesn't work well in biology for many reasons. But one of the main ones is that the flatness of biological systems is practically non-existent. You can have a cell and you can let it flatten out on a surface, but that does not give you representation, at least in our system, of what its biological morphology is. And so what we do is we coat our substrates that the cells are on for AFM testing, and have them maintain their rounded morphology so that the cell has a dome. And using the imaging component of the AFM, we come down with the cantilever, which in these cases is a bead that is on the same order of magnitude as the cell. So we're not using a pointed cantilever to get very specific, even further micro contact the way you would on a flat substrate like glass. But rather we are looking for the equivalent of like a bulk property of that cell. And we are indenting small magnitudes, but for AFM, they're not considered tiny by any, on the nanometer scale. We are indenting hundreds of nanometers, which for this kind of control of system is well within its capability. And by doing that and doing, modeling of that, we are then extracting these material properties. I have to tell you, it doesn't always give us exquisite control. We have tons of studies and experiments where we don't see differences. I find that the AFM analysis of different, for example, we've been collaborating with colleagues who are studying interfaces of bone to soft tissue, and they are asking us for their help to map out the different interface mechanical properties and things like that. We can see differences. Bone is hard, and this very other end of the tissue is soft, but what you'd imagine would be a linear interface ends up being not nearly as beautifully, different as one would imagine. And so when they want to make a replacement interface for their, engineering studies, it then becomes a little bit harder to target those material properties. So I think with the cells in this system, we do see some very fine differences because the effects appear to be large.
It's an
Not
amazing
subtle.
biohack. This qualifies as biohacking existing tools. It's
Thank you.
amazing. Thank you.
I will use that one day, biohacking.
Yeah.
Dr. Haglund.
Hi, Nadine. Very nice talk.
Thank you, Elizabeth. Nice to see you.
You know, I'm interested in TLR, so I have to ask a few things about that. But first of all, I would like to ask about, you know, your pain model. Have you tried to look at the grip strength, so actual discomfort in your model?
We have not tried to look at that in our rat studies. We currently have a different animal mouse model, which we're starting to pilot some of those kinds of grip strength and axial findings, and all really in great thanks to the studies that you've done in early on and looking in these models with the Spark null mice and with your collaborator, Laura Stone. So we're really inspired by the findings that you guys have and have tried to apply them to the mouse models, but we have not done them with our rat studies at all. We're somewhat limited in those other modalities. All the testing I showed you here and some of the other ones we've done in our own laboratory. We have a core facility that does mouse behavior, but they will not do rat behavior studies. So anything that we want to do on the rat, we have to develop ourselves or find a collaborator who can do it. Whereas the mouse, we have more abilities to try different things because there's a core facility that would help, you know, expand and leverage the capabilities there.
No, I have no idea how it would work in rats, and obviously it's Laura that developed all the behavioral tests that we have done. So I have no idea if it would be possible even to get the rats to grab on like the mice are doing. So another thing that I wondered, you use LPS in your experiments. There's some reports, depending on which LPS you're using, that it can activate both TLR4 and 2. Have you tried something like CRX 527 to be very specific to TLR and 4? And do you think it would make a difference?
So what was the thing that you mentioned, CX?
CRX 527 is a synthetic agonist that will specifically target TLR4, but it will not involve one of the co-receptors. But for sure it will, or for sure, what it's described to do is to be a pure TLR4 agonist. Yeah,
that's a great question. We have not tried it. I think our only insights into the role of TLR4 have been entirely based on these, blocking studies where we certainly have. I don't disagree with your point on that the LPS may be activating multiple channels or receptors, nor is HMGB1 only affecting one receptor, but that by using specific inhibitors, we're teasing out the contribution of each of those combination of ligand and receptor.
Another thing that we have seen is that TLR2, for example, is very sensitive to monolayer culturing, that you lose it quite rapidly in monolayer culture, but you keep TLR4 expression for longer. So, and I mean, in the end, it doesn't really matter if it's one or the other. You see the response when you activate a certain family of receptors.
Yeah, absolutely. And I think this really speaks to the importance of the toll-like receptor family as a whole. By no means am I implying that TLR4 is more significant than the others. I think they're all really important. They share, certain mechanisms. And I know you have a longstanding interest and body of work on the TLR2s. And I think we've talked about in the past about different model systems and how they have different contributions. I think we've been motivated by what we've been seeing in the patient samples that sort of drives our disease modeling, if you will. LPS is great in that it just turns on, and it is a hammer, as you saw. It does a lot. And so when we're in the beginning of studies and trying to figure out where's the best focus of a particular study, I do like working with the LPS model for that reason. But with the rat, for example, we don't know exactly, we haven't yet done the studies where we, let's say, give TAC242 and LPS and see if that changes anything. We've only done the simulation of the inflammation.
No, and it's possible that depending on which species you work in, you need to tickle one or the other. And still, like you said, the outcome is the same. The family of receptors are important, and you just have to figure out which one you need to start studying each species. And one short final question. You were talking about knocking out HMGB1. Yep. I'm just interested in which promoter are you going to use?
So the mouse that we are using, I think it's in the second promoter. It's a published study from Frank Schwab models in the kidney. I can send you the information about the mouse itself. We got it from him personally at Columbia, but it's also available in repositories, so it's publicly available mouse. And we're crossing it with Agracan Cree ERT2 mice to look at all Agracan expressing tissues.
Interesting. Thank you very much. Very nice presentation.
Thank you.
Any other questions? So one of the concepts that you're introducing is that one has to be aware of the loading history of the disc. For example, if I'm going to try to repair the disc to throw in stem cells, for example. So can you comment on that?
Yeah, so we've sort of over time become acutely aware that the response of the disc to therapy is not purely based on the biology of the therapy, if you will. And instead that the loading history appears to affect the response to therapy. We came to this conclusion by doing some studies using different but related model systems that I described here. And where we saw is that when you have physiological loading, so you take a whole disc, in our case, again, the rat motion segment, and we apply different loading profiles that change based on frequency or magnitude. And we found that some of these loading profiles cause the intervertebral disc to have mild to moderate pro-inflammatory activation, and other loading profiles have very severe pro-inflammatory activation. And that differs by static versus dynamic loading, etc. And so when we took those discs and co-cultured them with therapeutic cells like mesenchymal stem cells, we found that the response to loading affected whether or not the stem cells can act in an anti-inflammatory way. That's one of the presumed mechanisms of MSCs. So they can either differentiate and become a replacement cell, if you will, or can act by modulating the inflammatory microenvironment. And so in those studies, what we really came away with is that we don't think it's enough to just have the proper cell population that you want to use for your cell therapy, but that in fact you have to be aware and control the mechanical loading environment to promote the best, at least from a anti-inflammatory function, the response of the cells. And so what our goal is over the next several years, thanks to a new funding that we got to do this work, is to first do a number of series of studies in vitro to study the response of these cells to loaded discs, and then to in vivo implant the cells with or without controlled loading to see if we can push the therapeutic potential of these cells consistent with this hypothesis that the mechanical loading environment is affecting the cell's ability to be more or less anti-inflammatory.
Right, because you beautifully showed that when you have the hydrostatic pressure, you move it to 0.6 megapascal, you get the macrophages polarization to M2.
Right.
So in vitro, they show that if you go beyond 3 megapascal, you start getting catabolism. So
Right.
do you think you revert to M1? You go to M1?
That's a good question. So our system, our current system can't go much higher than 1 megapascal. So we're optimized for physiological loading with hydrostatic pressure. We don't have a control right now to do pathological magnitudes like 3 megapascal or higher. But Clark Hung's lab, I think, has a system that can go much higher than ours. And we could potentially examine that question and see what happens when you increase it further. Do they have sensitivity to those levels? It hasn't been done to my knowledge at those magnitudes. We did a fair amount of literature search to see if anyone has studied macrophage responses to loading with hydrostatic pressure. And there were some studies from, like decades ago, at very, very, very low levels of pressure on the single pascal, and I think motivated by lung studies. So the lung field has sort of tried to ask the question, does pressure affect, fluid pressure? Does that affect immune cells? Because certainly that's something that would be, they would be interested in. But the magnitudes they study are not at all relevant to the musculoskeletal system. And the findings there were somewhat inconsistent, like you saw all kinds of changes. So if we had to extrapolate further beyond what we're studying, which is the physiological range for the disc, and whether pathological levels push it in a different way, we don't know, obviously. I could speculate, but I don't know.
I just want to know which weights to when I go to the gym.
Well, if there's anything studying the spine has told us, is that our posture matters. So make sure you've got good form, right?
Yeah.
And short-term remodeling, inflammatory response to things like exercise are necessary, right? We can't eliminate all inflammatory response. It's the chronicity that's the problem. So
right.
So work on those things that strengthen your muscles and only causes a little bit of healthy damage.
Okay.
Not too much damage.
So
how strong are you?
So with your HMGB, one of the things I found in vivo with HMGB is that the redox state of HMGB is important because when it's in reduced form, it does good things. And then when it's a little bit oxidized, it starts to be detrimental. So what's the redox state of your HMGB?
It's a disulfide HMGB1. That's the one that we've focused on, which is the one that we believe to be most pro-inflammatory. However, our findings on the redox dependence, while in our in vitro cell studies seems to be very well controlled, when we move into the organ culture model, we see differences when we look at redox responses in organ culture, for example, when the ECM is present compared to just cells in tissue culture. And so that sort of suggests to us that there are potential complication or role for the extracellular matrix to play in those responses. And we don't obviously know what they are yet, but we are intrigued and have a hypothesis about that some the reduced form of HMGB1 might also be acting as a chemotactic factor. So it might be drawing in, immune cells and other things like that into a degenerated milieu, but that's still purely hypothesis at this point.
Right, that's great. Any other questions?
One quick question. Hi, Nadeen.
Michael.
Great talk and wonderful work as usual. I
Thank you.
really liked also your hydrostatic model. Very much interested in that. And I was wondering if you checked for any, played with the osmolarity in your cultures to see how that could affect inflammatory factors.
We have not. We've, stuck to a single osmolarity in our studies, and have just been tweaking the pressurization. But absolutely, it's an important variable, and that's something that, as you know, changes with disease, and certainly the literature shows that there's a lot of biological controls. So there could be further interactive factors and optimizations that could be done there to better understand the biology.
Thanks.
Thank you very much, Nadeen, for that really wonderful presentation. And thank you, and wish you all the success. I look forward to part 2.
Thank you so much. It's been a real pleasure to be with all of you. I really appreciate the opportunity, and it's wonderful to see you all in person. I see Lisbeth on the ORS spine section, but I don't get to see some of the others here very much at all. And I'm hoping that 2022 brings us all a more normative year, if you will, and that we can all, return to our normal interactions in person and our scientific community. And thank you to your experimental surgery department for having me here today.
Thank you very much. And thank you very much, everyone, for attending this seminar.
All right. Have a wonderful afternoon.
You too.
Bye.
Bye.